Today I will be drawing out and narrating the electron transport chain, also known as the ETC pathway in cellular respiration. The ETC takes place in the mitochondria, and more specifically in the inner mitochondrial membrane, also known as the matrix. As you can see, my two red lines represent the phospholipid bilayer. Above it is the intermembrane space, and below it is the mitochondrial matrix. Next, along the inner mitochondrial membrane, there is a series of complexes. The first complex I'll draw in blue, the second in pink, the third in purple, the fourth in green, and the fifth in yellow. The last complex is better known as the ATP synthase, which is actually separate from the ETC. Additionally, there are two other molecules in the ETC known as coenzyme Q and cytochrome C, which I'll draw in orange. Coenzyme Q and cytochrome C are also known as electron transport chain because that's where electrons get shuffled from one complex to another. Now that we have all the basic components, we can start with the pathway. So first, NADH and FADH2 arrive from the TCA, which is also known as the citric acid cycle. Then, NADH donates 2 of its electrons to complex 1. When NADH donates its electrons, the complex becomes supercharged, as shown by the yellow highlight, and has the energy to pump the hydrogen ions from the mitochondrial matrix to the intermembrane system at the top, which is also through the phospholipid bilayer. As it does this, NADH is oxidized to become NAD+. And through this pumping, more and more hydrogen ions are present at the intermembrane space, so you get this accumulation of energy, or the formation of an electrical chemical gradient, since there's more protons at the top than there is at the bottom. Next, complex 1 will pass its electrons to coenzyme Q. I just realized I made a mistake as I was editing this. It should be E- in the coenzyme Q and not E+ for the electron. At this point, we can go back to FADH2. FADH2 approaches complex 2 just as NADH did with complex 1. It will donate 1 electron to complex 2. So FADH2 is oxidized and becomes FADH. FADH2 is oxidized and becomes FAD. However, complex 2 cannot be supercharged, and this will not pump hydrogen ions to the other side, so it gets passed to coenzyme Q as well. At this point, the electrons are sitting in coenzyme Q are then passed to complex 3. Again, when electrons go from coenzyme Q to complex 3, it supercharges complex 3, which creates enough energy potential to pump the hydrogen ions from the mitochondrial matrix through complex 3 and into the intermembrane space. Again, this shuffling of electrons is creating the electrical chemical gradient at the top. Next, complex 3 will pass its electrons to cytochrome C. At cytochrome C, electrons arrive and then get shuffled to complex 4. At complex 4, the electrons enter and supercharge it, just like we've seen before. And again, the hydrogen ions are pumped to the mitochondrial matrix into this intermembrane space. Then, at complex 4, the electrons need to be passed one last time to the final electron acceptor, which is oxygen. The electrons are passed to oxygen, which splits into 2 oxygen ions, and hydrogen ions from the bottom are added to create 2 water molecules. Then we can move on to ATP synthase. Since there's a high concentration of hydrogen ions on the outside of the mitochondrial membrane, hydrogen ions will be pushed through ATP synthase. The protons will want to flow from high to low, so the hydrogen ions will flow back from the mitochondrial matrix, where then ADP is catalyzed to ATP energy, and this process is called chemiosmosis. And then this entire process, including ETC, is referred to as oxidative phosphorylation.